Hi guys, so I'm going to walk you through the biology exam paper, the higher tier one that we did in the spring, and just pick on some exam technique things we need to think about. And again, so I'm just going to pick up the bits that people found hard as we go through. So first one's about root hair cell. And how it asks you to label parts of the root hair cell. A little bit of a clue here because it looks like an unfamiliar cell. Bear in mind, a root hair cell is going to be found in a plant. So that should be a little bit of a clue first of all. Most people did fairly well in this question. Obviously working from the outside in, we're going to have the cell wall on the outside. There's slight confusion is it looks like there's 3 layers, one layer here, one here, and then inside. But bear in mind for a plant, this larger section will be the vacuole, and it pretty much fills the cell. And that question was done fairly well. The next question looks at how things get in and out of a cell, and as a recap, when something moves from a high to a low concentration, we call it diffusion. Now, a common misconception is diffusion only happens out to inside the cell. In fact, it happens equally well in the other directions. This is also diffusion because it happens from high to low concentration. Both of these other two, C and D, are going from a low concentration to a high concentration. So in both of these cases, we call it active transport. However, the question asks a little bit more than that. It says which way is the movement of oxygen molecules. So we're looking from a high to low, so that was going to be A. And movement of carbon dioxide molecules are moving again from high to low, leaving the cell, so that's going to be B. But don't be caught up that active transport and diffusion only happen in certain directions. It's about concentration gradients. Again, name and process of which these gases move into and out of the cell. We've said that already, is diffusion. Next one, I should look at some data, and so it says cells, tissues and organs are adapted to take in different substances and get rid of different substances, and it shows a concentration of 4 ions outside cells and inside cells. So just be really get your head in the table before you move on. So I've got the outside ones here and I've got the concentration here, and it might just help me to consider which way they're generally moving. Well, this way there is more on the outside and less on the inside, and it might just help me think a little bit about what's going on before I get started. It then says, use information from the table above to complete the following sentences. Sodium ions will move into the cells by the process of, so I need to find sodium. They're going high to low, so that's nice and easy. That's diffusion. Again, it's that concentration gradient. Potassium ions will move again into cells by the process of, okay, well, this time to go in the cell, they often go against the concentration gradient because it's more on the inside. So this one is going to be our active transport. Remember, that takes energy to happen. Okay, it then says some students investigated the effect of different concentrations, so there's the thing they're changing, of sugar in 4 drinks, A, B, C and D, on the movement of water across a partially permeable membrane. So they're measuring the water, so that's the thing they're measuring. Nothing about what they're keeping the same so far. Students made 4 bags from artificially permeable membrane, so that's quite important, it's about being artificial. They've put equal volumes of 5% sugar, so there's something they're keeping the same in each bag. And they've weighed each bag containing the sugar solutions. Here's the thing they're measuring, and they're placing 1 bag in each of the drinks, A, B, C and D. And after 20 minutes, something else to keep the same. They remove the bags containing the sugar solution and weigh them again, so weighing before and after. And the diagram shows how they set it up. Again, it's always important to just understand these experiments before you leap into the questions. You'll make less mistakes, and there's plenty of time to do so. So I've got the artificial bags, because you might have seen these, sometimes called Viscin tubing. They look like little mini sausages. And it's tied at both ends, and it's got sugar on the inside. So I'm going to annotate my diagram just to make you think about this. And then I've got 0.1%, 4.2, 6.9 and 10.6. So I might put more sugar on this one, some on this one, a bit in this one. It gives me an idea in my own mind. So there's more concentration on the inside. And each of them have got 5% inside. And it says what's on the outside, so that's 10.6, 6.9, 4.2 and 0.1. And that's just going to help me a little bit start to think about what's going on. Remember, this experiment's to do with osmosis, and osmosis is about the movement of water from where there's not much sugar to where there's a lot of sugar. So clearly in this one, we'd expect A to get heavier, B to get a little bit heavier. There's not much difference there, is there? C, actually we expect it to get lighter, and D, going to get a lot lighter because there's the biggest difference. The bag in drink A got heavier after 20 minutes. I'll explain why. So this is worth 3 marks, and this is about osmosis. So we need to say that water moves by osmosis. It's 1 mark. From, and I like the phrase sugar concentration, a low sugar, but solute is an even better word to use. So I'll put solute there, but I wouldn't expect you to put both. Concentration to a higher one. Now I'm going to make it clear it's higher. That's 2 marks, 2 statements. I've done said water's moving by osmosis from low to high. And in A, I need just to state where it is higher inside tube A. There is a higher sugar concentration. So I've got my 3 statements. I could jumble those around a little bit and put my last statement first, but I'm explaining the rules of osmosis and making it really clear where highest sugar concentration is. It then says, in which drink A, B, C or D would you expect the bag to show the smallest change in mass? Let's have a little look then. Well, we've kind of indicated that A is quite a big difference and D is quite a big difference. Without B and C, well, B has got the smallest difference, so water's going to move very little across there. It's going to be a small change. So it's going to be B, and it's the smallest difference in sugar concentration. Okay, well that's 1 mark, and what I'm aware of, it does say 2 marks, and there's already a mark given for my earlier reasons, so I need to give a second point. So what's going to happen? Less movement of water, and let's be really detailed by osmosis. So now I've really emphasised, I've made 2 clear points. And there's often where people fall down, they get 1 mark and don't expand their answer and drop marks. Right, next one says the image below shows some cells on the lower surface of a leaf. It doesn't look much like a leaf, but I've got these stomata and X. So let's look really careful what's going on here. So X is clearly pointing to the outside, a little bit less clear there, but I've got stomata clearly pointing to the black bits. So what the cells, okay, so it's about the cells on the outside labelled X called. Well, the guard cells are the ones that guard that opening and control whether they and help to open or close that gap. Palisade cells, remember they're involved in photosynthesis. And the mesophyll cells, they're the ones in the middle, and they of the leaf, and they have lots of gaps between them to let oxygen and so on in. Okay, right, then it says water is lost by evaporation from leaves is called transpiration. So you might remember you've done an experiment where you've got a plant and we watch the water move through and it leaves the leaf, and it obviously evaporates at the surface, and different things affect its ability to evaporate at the surface. Right, the student takes 2 leaves, A and B from the plant. So that's kind of, when we're going through this, start to critique it a little bit. They put Vaseline and grease on both sides of leaf B and did nothing to leaf A. So effectively, one leaf, they've covered it in with Vaseline, and the other leaf they haven't done anything to it. Okay, alright, they've written down the mass of each leaf and they attached the leaves onto a string as shown. So they've attached it there. And again, we've got the one that is covered in Vaseline and one that is not. They leave it for 48 hours, so that's the same in both, and they write down the mass of each leaf again, so measuring change. And they've calculated the percentage change in mass. Now it doesn't ask this, but that is so it's comparable because you've got one big leaf and one little leaf, a percentage change would equalise the relative sizes of each. It makes it comparable. Again, they haven't asked that today, but it comes up a lot. Give one variable their controlled investigation. Well, this is the point of highlighting as I've gone through. There's lots of things now. The time it's left for. Don't say time, let's fully spell it out, the time that it's left for. But I could have also picked out the fact as well. Just looking through what else I've written. So possibly nothing else other than it was from the same plant. But I prefer my stronger answer of the time it's left for. It's a nice number type answer. The mass of leaf A was 1.6 grams at the start, and after 48 hours it's 1.28 grams. Calculate a percentage decrease in mass over 48 hours. Right, remember to calculate percentage change, it is change over the original. So it's 1.6, take away 1.28. And I divide that by the original. That's the bit that takes a bit of thought. Well, the original was what it was at the start. So that is 1.6. So just simplify that. So that becomes 0.32 over 1.6 times by 100. So remember, change over the original times by 100. And whilst I grab my calculator, you probably already worked it out, equals 0.2. Now the question is, do I do a negative symbol here or not? Well, it's already said decrease, so that's okay. So 0.2 would be absolutely fine. Had it just said a percentage change rather than decrease, then I would have needed to put a negative symbol in there at the beginning. So just think through that a little bit. Does that seem roughly right? No, it's gone down more than 0.2%, so I didn't times it by 100. So obviously that gives me 20%. Okay, scrub that one out. And just again, does that seem sensible? Well, if I look, 1.2, it's lost about a fifth of its mass. So that answer seems correct. And it's worth doing that mental check first of all. Vaseline blocks the stomata. The percentage change in mass of leaf B was less than leaf A after 48 hours. Explain why. It's 1 mark. So what can't happen? Just look at the diagram again. So we know from before that water likes to leave through the leaf, through evaporation, and that can't happen. So evaporation, I don't know, it definitely didn't happen. So I'm just going to say was reduced. I'll be really clear which leaf I'm about in leaf B. And let's just develop it a little bit, and no transpiration, sorry, less transpiration occurred. Now I've kind of put 2 points there, but it's all about trying to make sure I develop my answer and make sure I really get the point. I've got 2 key words, evaporation and transpiration, in that answer. Right, what's going to give 3 environmental conditions that would increase transpiration? Well, what's going to speed this up? We've already seen it. Well, how hot it is. But lots of people just would say things like hot, higher. So I'm going to make it really clear: higher temperatures, a higher wind speed, because that reduces humidity, and also to go with that same idea, a lower humidity. And it's all about when evaporation takes place. We've got lots of particles in one place and they want to move into the air. If there's lots of particles in the air, it's harder for that change to take place, and so particles can't actually evaporate. If we have a big wind going by, it blows away the particles. Or if there's less in the air because less humidity, it blows away the particles. And finally, when there's more temperature, there's more kinetic energy, so they're more likely to leave the liquid and move into the air. Right. Plants absorb light to photosynthesis. Most people do really well on this one. Okay, if you had it back to front, plants, think about it, they need water, they need CO2, and so that gives them, they then make oxygen glucose. Figure 1 shows some of the apparatus that can be used to measure the rate of photosynthesis. And this is an experiment that you probably have seen, done a photosynthesis topic. There's our plant, it's given with little bubbles of oxygen, and at the top we've got a volume of oxygen starting to appear. It says the rate photosynthesis in pondweed is affected by different colours of light. Describe a method you could use to investigate this. And you should include what you measure and what you can control. Now I've shrunk the gap a little bit here on the printout, but this was done sort of okay and then very well by some. And to keep it very simple, it's about writing in bulleted sentences. So what am I going to measure? Well, I'm going to measure either the number of bubbles, but it's not just the number of bubbles forever. I need to say in a certain amount of time. So I'm going to say 1 minute. It's okay for you to suggest times and volumes of things. What am I going to control? Well, I'm going to control the same, again, just to say same pondweed is not great. The same length of pondweed is much better. And we also have already said other things that affect plants, such as temperature. So I'm going to make sure my temperature is the same. And again, develop my point. How do I do that? Using a thermometer. Now remember, thermometers don't actually control temperatures of things, they just monitor it to monitor. Okay, so I've got 3 points. What am I going to change? Well, it does say different colours of light, and there's different ways you could do this. Some people suggest different colour light bulbs, that's fine. Some people suggest different filters. So cover beaker in different, sorry, handwriting's going to be messy, coloured filters. Again, this is, I've got 5 points now. So, and then shine light on pondweed. Not totally convinced that's really mark where you point. Ensure it's the same distance each time. So in the same distance in pondweed. And finally, I've only got 5 points. Repeat each colour 3 times and calculate a mean. And so I've now definitely got 6 points. I could give a little bit further detail as well. I can mention how I control the carbon dioxide concentration using bicarbonate soda, but I've got my 6 points, and really to get to 5 or 6, if you can do that, that's a really big improvement for many people. A typical answer is about 4 marks there. If you didn't put controls, you didn't get more than 4 points. It was capped out, and if you can always remember controls. Right, so scientist carries out a similar experiment and have results from figure 2. So rate of photosynthesis, and it's similar, so be careful, light intensity. So always remember these graphs, that as you increase in light intensity, what's happening, so the rate of photosynthesis is going up until about here, when actually no change takes place because temperature or carbon dioxide now become limiting. And most people make the mistake of just saying overall that temperature or CO2 are the limiting factor. They are on this side, they're not here, because clearly adding more light increases how much photosynthesis happens. So it says, science says light stops being a limiting factor at light intensity of 20 units. Give evidence from figure 2 to support this statement. You needed to say at 20 units. That's where people drop marks. They didn't say from this point on, but from this point on you can see that there's no increase in light in photosynthesis with an increase in light intensity. Most people did alright in that, but not everyone said 20 units and so dropped marks. What could be the limiting factor? At 25 units, we've said it already, carbon dioxide or temperature. The next one's about human activities in our ecosystem. And the graph shows the volume of peat compost and peat-free compost in gardening from 1999 to 2009. Again, just look at the graph. What's the general trend? Well, it looks fairly flat. Don't worry about little fluctuations, but do look at significant changes. And then this one is a steady increase. Volume in thousands of meter cubes. So describe the trend shown in the graph. Again, most people did okay, but some people didn't state that it was in 2005 that we saw, or from then that we see a decrease. Just to say peat goes down is not true. Peat compost doesn't go down, does it? Okay, it's steady for the first part. So include data in your answer. Saying that it's rising from 1,700 to 4,000 is a much better answer. But don't just give numbers, include the words as well. Sometimes there's merit in saying it's doubled. In this case it hasn't, so I wouldn't include that, but if there's can get language like that, that's good. What effect does the destruction of peat bogs have on the gases in the atmosphere? Well, it increases CO2. I know many of you haven't covered this topic yet, but it's actually linking into things you should commonly know anyway. And deforestation is damaging ecosystems. Well, the main reasons is habitat, through things like soil erosion, and so on. Again, that was answered well. Okay. At the seashore, the tide becomes in and out twice each day. Some students investigated whether 2 different species of seaweed could live only at certain positions on a rocky shore. Seaweeds are plant-like organisms that make their food by photosynthesis. So this question's on the rocky seashore, and it shows 2 species of seaweed that students investigated. And typically in an exam, you probably haven't heard of bladderwrack, and you probably haven't heard of sea lettuce, although they are the most common types of seaweed you'd see if you went down many kind of rocky beaches. But look at the information given, because every bit of information the examiner gives is there for a reason, and that's really important. So it says air bladders. And again, there's these little air bladders on there. You might not know quite what an air bladder is, but a bladder is like a sack. You get a drinking bladder. Sometimes you take out for runs or cycling, and this one obviously contains air. Nothing particular about the sea lettuce other than it's particularly thin from the drawing. And then it also says it's got this thing of the hold fast, which fixes it to rock. So seaweeds don't have roots because they're not quite plants, but they do have to glue themselves to rocks so they don't float away. Right, it then says the students placed a 50-metre tape measure on the rocks at right angles to the sea. So there's no other information given. It helps to visualise what this is. So here's my sea down here, and what they've done is they've put a tape measure that's 50 metres long at right angles to the sea. And they've placed a quadrat next to the tape measure. So it might help you if you're particularly stuck with these kind of questions to visualise it and draw it out. And then recorded whether each species was present or not. So they've gone through and probably put a tick or cross for each of those 2 species above. And they repeated it every 3 metres down the shore, so they can get a bit of a pattern. So this is a transect rather than random sampling, and they're looking for a gradual change as you move away from the sea. A little reminder that it gets more, actually bizarrely, it gets more salty the further away from the sea you go, because the salt is left behind as water evaporates. So water actually becomes more of an issue as you come away from the beach. And so repeat every 3 metres. Right. Figure 2 shows a section of the seashore and the student's results. So this again will help you visualise what's going on. So I've got 50-metre tape measure that runs the length of the beach. They've got little pools of seawater, and clearly as it gets higher and higher, even at high tide, remember, because the sea comes in, some parts of the rock will never be exposed. Right, and it shows the results they've got. So these type graphs, sometimes called kite diagrams, show where the bladderwrack is present. So it's present from here to here, and the sea lettuce is found here and here, here and here, and here and here. Notice it seems to coincide only where there's water. Right, again, got my headwind results, so I'm ready to move on to the question. The students placed the quadrat at regular intervals along a transect line rather than placing the quadrat at random positions anywhere on the rocky shore. This is a particularly hard one to word and to explain, but we're really looking for the fact that we are not doing random sampling. And from this one, that we are trying to show the effects of a particular abiotic factor. So that's why we're going to be using a transect. Okay, and the point of it being a transect is transects show how the organism is positioned in relation to the sea or to that abiotic factor. And that's the difficult one. It's quite difficult to word that one. So again, when you're looking at mark scheme, that phrasing, its position in relation to the sea, is really important. I.e. that we've got, along our beach, we know that our particular organisms might occur at certain bits, and if they're in particularly high abundance here, we know that that's because of a certain distance away from the sea. If we did random sampling, let's see random sampling, different colour. When we randomly sample, we may not actually get organisms in certain positions. We wouldn't really know if there's an effect of the rocky little pool of seawater, for example. So it's important to make sure we do a transect when there is a gradient. Okay, next one is how can institutes improve their investigation to ensure they produce valid data. Okay, this is really important. Valid data is about ensuring they're controlling all the variables. This is not my answer, this is what validity means. Controlling all the variables. And in this case, we want to be certain that we haven't just kind of sampled it once and just got this idea that it's just a single sampling. So although validity doesn't always refer to reliability, they would certainly want to make sure that their conclusions are true. And in this case, they repeat at different transects or different positions on the seashore. And again, if they are in other contexts, when you talk about validity, we'd also consider what they're doing to make sure all variables are equally controlled. Okay, repeat the data. Students concluded that bladderwrack is better adapted than sea lettuce to survive in dry conditions. What's the evidence for this conclusion? So again, evidence means, and it says it twice, use figure 2. So it says that bladderwrack is better adapted than sea lettuce to be in dry conditions. Well, it's 2 marks, and I would say that we've only got, as we pointed out already, sea lettuce when there's water. So sea lettuce is only present in water, or the little rocky sea pools. And it's 2 marks, so let's be dead clear, whereas bladderwrack is further from the sea. And I want to be clear, than sea lettuce. And I'll go and say, which obviously it's further from the sea, there where it's likely to be drier, or there's less water. So I haven't just said what some people said is it's drier or bladderwrack's in dry conditions. I've made it really clear. It's about being really clear that it's further from the sea, and we only find sea lettuce in the sea itself. So 2 bits there, and develop both answers. And that's really important to gain the marks. Bladderwrack has many air bladders, and they help the bladderwrack to float upwards where the sea covers it. Suggest how this helps a bladderwrack to survive. So just think about it, it's in the sea, we've got our bladderwrack near the surface. What's near the surface? Well, it's the photosynthetic, isn't it? It's a seaweed, so there's more light. 2 points to help it photosynthesize. Some of you said to get more carbon dioxide. That was great, but a lot of people missed out the idea that seaweeds photosynthesize, and therefore it's about being nearer the surface to be able to photosynthesize. Right, please use some of these ideas and just write yourself down 2 or 3 points. You think, well, I didn't approach the question that right way, I could have structured it differently, so you develop exam technique. I'm sure there's bits in here you've done already, but I want to focus on the bits you haven't done and how you can improve. Well done, guys, thank you.